Are you at your desk in a focused, distraction-free place with a pencil and paper, trying these? All right, this one's from 2016 CDT release. These are this state released practice questions for their for the test. So go ahead, pause the video and try this. All right, let's see how to attack this one. Part A, um, cellulose. Right, I noticed that OSE. That's a carbohydrate. And you should just know cellulose is a polysaccharide. It has hundreds and hundreds of these um, glucose monomers, and they're found in plants. And they make up the cell wall of plants. All right, it's a polymer. That means it's made up of many, many monomers. One of those would be a monomer, which contains repeating units of monomers. Glucose is the monomer, so that is glucose. Right, this whole thing is starch. Just part of it. It's actually hundreds and hundreds of units long. All right, what type of reaction helps form, there's the key word, form the bonds. All right, so what helps makes these bonds, connects monomers to polymers? That's dehydration synthesis. You don't see the full name here, but the answer is D. This can also be known as polymerization, so I'll write that down. Or condensation. All right, reactions. So, but most likely you'll see dehydration, synthesis. All right, part B, describe how water is involved in this reaction. Okay. So, guy, draw. This was one of our note checks. Let's use glucose as the example. So, in each of these monomers, there's an OH group and then an H on either side. And so, let's just make two glucose monomers. Okay, and you have countless of these. All right, and how are they going to turn into this larger polymer? Okay, so the question here is, how are we going to make this bond? How are we going to connect these two and make it look like a larger molecule? Right, well, the way that happens is an OH from one monomer is going to be removed, and H from another monomer is also going to be removed, and that's going to form a water molecule. That's why it's called dehydration or condensation. Okay, and in that process, when those are removed, then a bond can form between the two. All right, with H and OH there, a bond can't form. So those have to be removed, and then the bond can be formed. All right, so that's part B. How is water involved? So the answer is water is removed. So let's add that. Part B. Or you can say water is created in order to form the. Okay, so there's part B. All right, let's take a look at part C. Give another example of this type of reaction. So in the three types of the three molecules that are formed this way are carbohydrates. We just saw that. Carbohydrates form through dehydration synthesis. Another one is proteins, and the other one is nucleic acids. The only one that doesn't of the four that we learn are lipids. Lipids don't have monomers. Okay? No. But all three of these other two can also form the larger molecule through dehydration synthesis. All right, so let's take a look at how proteins are formed. All right, proteins are formed of amino acids. All right, they kind of look like this. Not really. You can okay. This might be a little more accurate. Okay, so they have hydrogens and other atoms too, but I'm just going to draw generically here. So again, you have an OH on one. There's another amino acid. There's an H on another one. Okay, and in order for us to combine these amino acids to form a protein, or in this case, more accurately, a polypeptide, in order to form that bond, right, we have to remove an OH and an H, and that's going to create water and replace it with a bond. So there's your answer to part C. Okay, let me read the question again. Give another example. All right, so how do we put that in words? All right, part C. Okay, another example of dehydration synthesis. Amino acids, oops, a hydrogen and a, call this hydroxide or OH. You can just write OH. Now, polypeptides are what proteins are before they are folded. But you can say proteins, okay? And that bond specifically is called a peptide bond. All right, so this here is called a peptide bond. All right, it's a little bit more complicated with a nucleotide, so I would go with this example.